Just a reminder to keep an eye out for the newsletter from UK-ICN for news about the network and upcoming events, and there should be news of a new website at some point in the future as well, which is exciting. So so we've got two great speakers today. I'ah Donovan Banfield, PhD student from University of Liverpool, and Professor Rich Stanton from Cardiff University. So first off, we're going to have a talk about the characterization of SARS-2 genomic variations in response to molnupiravir treatment in the Agile phase 2A clinical trial from I'ah. So I'ah, are you okay to try and share your screen for us? And the talk will be about 20 minutes or so, 15-20 minutes. There'll be opportunity for questions after that, and then we'll go over to Rich's talk after that. Okay, I can see that fine, I'ah, so whenever you're ready, you can begin.
Okay, cool. Thanks for the introduction, Dan, and I'm excited to give a talk to everyone here today. I've kind of put a shorter title on the slides just for ease of reading, but yeah, essentially, this is the work I've been doing for the last couple of years as part of my PhD, and we were investigating the SARS-CoV-2 genomic stability in response to molnupiravir treatment in humans. So I'm sure everyone on this talk today has seen this slide in some format or the other, so I won't go into too much detail, but it's basically just to make a point that SARS-CoV-2 has a relatively complex life cycle, and it requires the manipulation of multiple different cellular factors and structures. And that also means that there are multiple steps at which antivirals can be targeted. So we have, actually let me get a pointer up. Yeah. So we have entry, where you can have protease inhibitors and also monoclonal antibodies can block entry. Endosomal ossification inhibitors, which mainly I believe are sort of lab-based inhibitors. I don't think there's any on the market treatment in humans. And then also we have the protease inhibitors, intracellular protease inhibitors such as Paxlovid, and also replication inhibitors such as remdesivir, favipiravir, and the like. And so one such antiviral, molnupiravir, acts in this category here as a replication inhibitor, and as we would discuss, will be the focus of my talk. So molnupiravir was originally developed in its current formulation as an antiviral against influenza. Previous iterations that were less orally bioavailable were initially developed against Venezuelan equine virus and several other RNA viruses. It's administered as a prodrug, so it has a chemical group attached to it that makes it more orally bioavailable. It gets metabolized in the cell into its active form, which is a molnupiravir triphosphate, so where it acts as a free nucleotide to be incorporated into the nascent RNA, and it can stably bind to either guanine or adenine, and it seems to get away with this, that sort of it being incorporated as a different base isn't recognized by the viral replication proteins. And in doing this, you get an accumulation of mutations, G to A mutations and C to U mutations, which are described as transition mutations. And this induces lethal air catastrophe, has been described before. So I'm just going to go over some early molnupiravir efficacy data, which is quite old now, I guess. Sort of the first trials were in 2020. Which is crazy, about 3 years ago now. But so molnupiravir was repurposed for COVID, and clinical trials began at the beginning of 2020, at the end of 2020, sorry. And the first major clinical trial was this MOVE-OUT study in the US. And they showed that there was a modest benefit, there's 3 percentage point difference in incidence of hospitalization or death. And this was given the time that it was conducted in, mostly on unvaccinated individuals. And since then, we've had several regulatory approvals. So the UK's MHRA, the regulatory approval body, gave temporary authorization in November 2021, based on pretty much solely based on this MOVE-OUT study. And the US based on the MOVE-UP study as well, gave emergency use authorization. And the EU, I think, when I originally written these slides, they were still assessing authorization, but I think they have declined to give authorization as it stands. So yeah, so this study had unvaccinated participants. It was 3 variants of concern, and they did not share any genomic analysis of the virus before and after treatment. So that means we have 3 remaining questions. What are the clinical benefits of molnupiravir in vaccinated individuals? Is molnupiravir effective against variants of concern? And can we identify any treatment-associated mutations that might lead to resistance? And the work I'm going to describe today has been out in these 2 publications towards the end of last year, and they are both open access. So the outline of this study, so the study was conducted in collaboration with the Agile Coronavirus Drug Testing Initiative, which is a UK-wide initiative for the rapid evaluation of treatments against COVID-19 specifically. And this trial, the molnupiravir trial, was the 2nd candidate-specific trial. And as you can see here, there are several inclusion criteria: said to be PCR positive, over the age of 18, and within the first 5 days of your clinical symptom onset. Patients were randomized. It's a double-blind randomized controlled trial, so to either placebo or drug. And then patients were sampled regularly up until 29 days. And the main outcomes were time to negative PCR, reduction in disease progression, and reduction in hospitalization and deaths. And there are around 180 patients included in this. And so the outcome that we were measuring was the exploratory secondary endpoint, so the genomic evaluation of SARS-CoV-2 after treatment. And so what we did is so the patients are getting swabbed regularly. So we took some of the RNA that was extracted from these swabs, originally intended for PCR confirmation, and we used the samples from days 1, 3, and 5 to go on into our whole genome amplicon sequencing pipeline, which is very similar to the Arctic sequencing pipeline, but we used a kit called ETC SARS-CoV-2 WGS, which works in a very similar manner, but it's just designed to be easier to process and specifically for use with Illumina sequencing. So the genome selection criteria we had, so this is just a brief overview of the bioinformatic pipeline that we used. We used the sequencing kit that we used nicely came with its own pipeline to get from your raw data to your sort of consensus output and your lineage assignments. But we also used another tool called Diversity Tools and several other custom R packages to have a look at the minor variant analysis. So the consensus genome is just the most numerous mutations that are there that build up the whole consensus of your viral population. And the minor variant analysis looks at all the variation underneath that major variation. And so we looked at the nucleotide variation and then also the predicted amino acid changes that would happen as a result of the nucleotide changes. And so we also identified several different lineages. And as you can imagine, because it was from sort of late 2020 until mid 2022, we have a whole variety of lineages here. So all the way from Alpha until Omicron as well. So the first question, what are the clinical benefits of molnupiravir in vaccinated individuals? The short answer is modest, maybe not even worth it. Hopefully there's no pharmaceutical people on this call, but basically the results showed that molnupiravir is only marginally better than placebo in all individuals, and there was a slightly better benefit for unvaccinated individuals. And the overarching conclusion from the clinical conclusions was that they had preset a probability of 80% for whether 80% likelihood that molnupiravir was more effective than placebo as a threshold for recommending a larger study, which molnupiravir did not meet. So the other question was, since we had samples from those different variants of concern, is molnupiravir still effective against these as compared to the first trial results where it's just sort of Wuhan backbone? And we can see generally molnupiravir reduces the risk of hospitalization for pretty much all of the variants of concern besides Delta. And also Delta was the most numerous group that we had as well. So maybe this is potentially more accurate than some of the other groups where we had very small sample numbers to do such an analysis. And as I mentioned before, the likelihood that molnupiravir was superior to placebo was only 75.4%. And so it did not meet the self-imposed threshold for recommending molnupiravir for a larger scale study. And then the third question, can we identify any treatment-associated mutations that may lead to resistance? Which I think is definitely the question on everyone's mind when you're administering a mutagenic drug for a virus. So essentially, after some of the stringent sort of genomic quality controls were put onto the data, we had roughly 65 patients remaining in the placebo group and 59 patients remaining in the molnupiravir group. And so that small subset is what we're carrying on the rest of the visualization for. And these, basically, we detected the hallmark of molnupiravir treatments, as I mentioned at the beginning, where you have these G to A mutations and C to U mutations, and that can be expressed as a transition mutation to transversion mutation ratio. And so you can see in green here is placebo, where you see the transition ratio stays fairly stable across the first 3 days of treatment. And as you can see in molnupiravir, that it increases over time. And the difference between the two groups is significant at day 3 and day 5. Now, breaking down those mutations into the individual parts, G to A, we see does increases the same way, and C to U as well. Interestingly, A to G, the reverse mutations are less likely to happen, and the U to C mutations are at a very, very low level, but still significantly significant between the two treatment groups. So in conclusion, the mean transition ratio of SARS-CoV-2 is significantly increased over time in molnupiravir patients, but not placebo. So having a look at the actual nucleotide mutations, we wanted to see if we could look at the predicted amino acid profile and see what sort of changes are happening in the monovariant space. And so again, we have placebo on the top row and molnupiravir on the bottom row, and you'll see that this dotted line here at 50 proportion of 50% is our monovariant threshold. And as we can see for Delta, we see that you have an increase in minor variation across time, but you see it more so in molnupiravir. And so another interesting point to make is that generally in genome diversity estimations, the higher your viral load gets, you can see it, you tend to see an increase in minor variation, because you have less virus there in the first place. But we still see a clear difference between the two groups. We see the same thing in Omicron as well. And sort of the question that we wanted to ask, especially when we would see mutations crop up against things like Paxlovid that have a very specific amino acid substitution that leads to resistance phenotype, we thought we should look and see whether there are any selection pressure on the viral genes that we might expect to be affected by a replication inhibitor. So that would be NSP12, which is the polymerase, and NSP14, the proofreading enzyme. And we have the data here just for Delta and BA1, but we didn't really see any particular hotspots of minor variation at any point across those two proteins, which is a good thing to hear or see. And then this is just another interesting observation we had out of our study, because with a lot of genomic analysis, especially when you're using national or international databases, a lot of the genomes that are deposited are just the consensus genome that I mentioned before, so just the top level information of that virus population. But what we could see in our data, because we're looking at the minor variation, is we saw these interesting patterns in Delta and Spike. So Delta had these two positions that had a mixed population. So the dominant mutation there wasn't entirely fixed in the population. And we saw the same with BA1. And when I looked at these positions and what those mutations were, I found sort of an interesting story. So for Delta, we saw this T19R substitution in Spike, And that site is actually a VOC site. So that's a lineage defining mutation for Delta. And it's interesting to see that even though over time that evolved to be the amino acid that changed, it's not entirely fixed yet. And I guess at these stages, it was sort of right in the middle of the Delta outbreak. So it's interesting that maybe these sites are quite flexible in the mutations that they allow. And the same interesting story of BA1, we saw at these two positions, T20 and P26. Actually, they are not BA1 mutations. They are lineage defining mutations. They are P1 lineage defining mutations. And it's interesting that they are in the some of these BA1 patients as a minor variant change. So this basically just to illustrate that besides detecting molnupiravir-induced transition mutations, our deep sequencing and intrahost variation analysis has allowed us to identify some positions within the spike gene that seem to have persistent populations, particularly in the Delta and BA1 lineages. So to conclude, this was the first confirmation of the mechanism of molnupiravir on viral replication in humans infected with SARS-CoV-2. We did not identify any amino acid substitutions that were enriched consistently at specific sites in molnupiravir treated group, including in NSP12 and NSP14. Intrahost variation analysis identified the persistent subpopulations in VOC sites of Delta and novel sites in BA1. Just to say that further surveillance of SARS-CoV-2 after the rollout of molnupiravir, especially in countries where it's been given widespread approval, is required for monitoring potential resistance. And some future work that I have planned is I want to characterize some samples with that had elevated viral load at later time points. Just to say this was in both placebo and molnupiravir treated patients from this study. And we also have access to some phase 1 data from Agile that was the dose escalation study. So we can investigate the effect of subtherapeutic doses on molnupiravir. And I think there's been some news, at least on Twitter, of these clusters of molnupiravir or clusters of sequences that have been deposited that have a molnupiravir-like signature. And so it'd be interesting to see what the persistence of molnupiravir mutations are after treatment. So once a patient finishes their treatment, if they do have molnupiravir mutations lying around, how long, if that virus is still competently replicating, how long are those mutations maintained in the population, and do they remain there after being transmitted? And also to look at the effect of molnupiravir on monoclonal antibody neutralization. I think monoclonal antibodies have been one of the most vulnerable sort of countermeasures we have against SARS-CoV-2, especially with the evolution of the variants. And so it's interesting, it would be interesting to see whether we give, when we give a mutagenic antiviral, whether that compromises the effectiveness of monoclonal antibodies. And with that, I'd like to thank everyone in the Hiscox lab and also the collaborators at Agile, which are honestly way too numerous to name all of them here. And a big thank you to the team at CGR for helping us sequence these quickly. And thank you for listening.